How does electron movement through cellular respiration actually result in ATP production? You may already know that reduced electron carriers like NADH and FADH₂ carry high energy electrons. But how does that actually result in the production of ATP? Electrons never touch ATP synthase. They never touch ADP or inorganic phosphate. So how does this actually result in a transfer of energy that allows us to finally do cell work? Well, I'm Miss Parrott, and in this video, we are going to be following the electrons in cellular respiration to answer that very question. So first, where do these electrons come from? Initially, they come from glucose that is either consumed by a consumer, absorbed by a decomposer, or made by an autotroph. So, and the electrons are the negatively charged particle in all these H's right here. But glucose doesn't give electrons directly. There are a lot of intermediate reactions, and so when glucose breaks down into smaller molecules like G3P, pyruvate, and others, those smaller molecules are going to be the ones that hand off their electrons for, these are the electron donors during cellular respiration. So the electron donors, like G3P in the case of NADH, G3P gets oxidized. Remember, oxidation is losing electrons. So G3P transfers 2 electrons and 1 proton to NAD plus to form, so when these get added, then this NAD plus gets reduced to form NADH. Check out the previous video for all the down low on that. So we are forming reduced electron carriers like NADH and FADH₂ by oxidizing glucose derivative molecules. But then what? So electrons are then going to enter the electron transport chain. So let's imagine we are in the mitochondria. So here's my purple peanut, that's the classic mitochondria, with the outer membrane and the inner membrane. We're going to zoom in here on the inner membrane, that's here. All right, so electrons will enter, this is called the electron transport chain. That is a series of, I've drawn them here in blue, of proteins whose job it is to transfer the electrons. And we're going to break it all down. Like, if you were ever confused by the electron transport chain, this is the moment where it's like, I get it. Okay, so NADH and FADH₂, those are our reduced electron carriers. Reduction is gaining, so they have the electrons. Then they are going to carry them to these blue proteins, which have electron acceptors on them. Okay, so these electron carriers are oxidized, so they're going to lose their electrons. So see, I have in green the reduced form, then they're giving 2 electrons to the electron transport chain to form their oxidized form, right? So NADH drops it off at this first protein. FADH₂ will drop its electrons off here at the second protein. And these electron acceptors, these blue guys, are now reduced because they have gained, reduction is gaining, gained the electrons that were brought to them by our electron carriers. All right, now NAD plus and FAD are going to get recycled. So they're going to go back to other parts of the mitochondria or of the cell to get new electrons and come back to the electron transport chain. Then electrons are transferred through the electron transport chain. So check it out. You can follow this little red path So the electrons dropped off by NADH are going to go up here through this first protein, across CoQ, down to the third, across cytochrome C, down to the fourth, and come out here, right? And then at FAD, they're going to go in the second one up to CoQ to number 3, cytochrome C, protein 4, and down. So those are going to be moving. Now, what's kind of fun here, so because there is nowhere in the AP Biology course exam and description that says you have to memorize the proteins in the electron transport chain. And I'm not saying you should, but cytochrome C, that's this protein right here, it will often show up in the evolution section or in questions on the AP exam about evolution. Because, right, it's a protein, which means it's coded for by DNA, and organisms with more similar DNA are said to have a more close ancestry, a more close common ancestor than ones with more different. And so the cytochrome C protein, the DNA or the amino acid sequence for that, is often used to compare evolutionary relationships. So don't be surprised if you see that. Also, CoQ is like, it's even sold as a supplement now. It's called CoQ10. It's the same thing here, ubiquinone, and you can like buy that at the CVS. So crazy. So all this is real. That's the thing. I feel like when biochemistry that I forget is that it's so tiny, it's so molecular, it's so diagrammed out and alien to our lived experience that we forget, like, oh no, this is real. And it shows up all the time. All right, let's keep going. What happens after the electron transport chain? So, well, before we get to after the electron transport chain, like, what is the point of the electron transport chain? So look, if you look at the final net reaction of cellular respiration, it's like glucose plus oxygen yields CO2 and water, which is a classic combustion reaction. But if we combusted glucose, boom, all at once, that would release lots of energy, heat, and light, and that would be dangerous. And we do not want spontaneous human combustion happening. So we have these small, many intermediate steps to release energy a little bit at a time to make cell work happen. So check it out. If you recall from the previous video, we are like electrons start in this system at a higher higher potential energy. So here's your electron. This is the higher potential energy. Here's the lower potential energy. And as these electrons move from one step or like one protein to the next, you can see I've numbered these steps with the same proteins that are in the electron transport chain. So as they enter this, as they move from one protein to the next, they're moving to a lower potential energy, And so as they move, remember, it's the movement itself. That movement allows the energy to become available for cell work. So, right, as electrons move through the electron transport chain, potential energy is going to decrease, and then the rest of the energy becomes available to do cell work. Now, some energy is released as heat because our metabolic systems are not 100% perfect. They're not 100% efficient, not remotely. They're about 10% efficient. So some energy is reduced as heat, and yeah, you can feel that on yourself. Hey, now recall one of the biggest ideas is it's that as electrons are transferred, so as they are moved from one place to another, that is going to create an energy shift. So what are we using this energy for? Well, we use that energy from movement of electrons to move protons. So let me just lay out again, we're back at the inner mitochondrial membrane. Up here is the intermembrane space. So that space between the inner membrane of the mitochondria and the outer membrane of the mitochondria. And then down here on my diagram is the matrix, which is this the most inner part of the mitochondria. All right. So again, here's our electron transport chain of proteins. Now you can see I've added the protons. So we have protons in the matrix, we have protons in the inner membrane space. So energy, so big E stands for energy, and little e minus stands for electron. So if you're writing this out, just so you know. All right, so energy from electron transfer, right, going from one of these blue blobs to another, which it's not a blue blob, it's a protein that actually like does stuff. It's used to move protons from the matrix, from this inside, into the inner membrane space. So what this does is create an electrochemical gradient, which is a difference in proton concentration between the inner membrane space and the matrix. It's also going to create a difference in charge. Now, while these are protons down here, and they're positively charged, and these are protons up here, and they're also positively charged, because there are more protons, a higher concentration of protons up here in the inner membrane space, this is considered more positive relative to this space down here. So this gradient, that is essential to the next step. So this is why we've got to have electrons doing their job, is to pump protons through. So not only are these oxidizing and reducing to pass electrons, but as the electrons move through, certain channels, certain pathways are being opened. Hydrophilic pathways are being opened in these proteins to allow the protons to pump through and not come back out. So, because otherwise, right, the inner mitochondrial membrane is a lipid bilayer, just like the cell membrane, and charged particles can't just pass through there. It's nonpolar, so it's like no charged particles, ions. You stay on your one side or the other. Polar stuff stay on one side or the other. So that's why we have to have these proteins to allow the protons to go through. All right. Once we have our gradient, before we have our gradient, right, the equation for cellular respiration, last time I checked, was C6H12O6, glucose. This is the net at the very end, right? Plus oxygen makes carbon dioxide and water. So what's the point of oxygen? Where does oxygen fit in this picture? Oxygen fits in as the terminal or final electron acceptor. So in an AP biology exam, you will see the word terminal, like terminal electron acceptor. But in a textbook, you may also see final electron acceptor. So that's why, but they mean the same. It's the one at the end, right? So, right, our reduced electron carriers give electrons to the electron transport chain. They pass it through. Then it's going to get here to number 4, the fourth one, the fourth protein. Now, this is what's super important. This isn't just a tunnel or like an electron front. It's not just like there for electrons to go through. It's actually an enzyme. So it like functionally does stuff too. What it's going to do is take O2, which is 2 oxygens bound together by a double bond. And where does this come from? either breathing it in, or if you're some other kind of non-breather, right? Diffusion, oxygen is diffused into your cells. And what this protein, what this enzyme will actually do is, if there's an enzyme mediated breaking of the double bond, right? So now, if that double bond is broken, now there's electrons to be accepted. So then this enzyme is going to add 4 electrons and 4 protons, and the protons are coming from the proton pool here in the matrix, to make 2 water molecules, right? So we have those 2 oxygens and then 2 hydrogens, which are made up of 2 electrons. Like, 1 hydrogen is made of 2 electrons and 2 H2. That's 4 hydrogens. See, the math has got to math. All right, 4 total hydrogens come from 4 electrons and 4 protons, 1 electron and 1 proton each. So just remember where these reactants are coming from. We get oxygen from breathing in. We get our electrons from the reduced electron carriers like NADH or FADH₂. And then we're getting our protons from the proton pool in the matrix. And then that's where we get water as a product of cellular respiration. It's because O2 is the terminal acceptor. But what if we stop breathing? Or what if there's no oxygen available in the atmosphere? What is that effect? If you have no oxygen, then there's nothing to catch the electrons coming out. So they're not going to come out, right? Because remember, this isn't a tunnel. It's not like it just dumps out. This is actually going to be, it has to take the electrons that are in it and attach them to an oxygen. So if there's no oxygen there, it's not going to attach the electrons to anything. Then you start to get electron backup. So there's no continued flow. And if there's no electron flow, right? Electron flow, the whole point of that is it is that energy, as those energy shifts change, that pumps the protons into the inner membrane space. So if electron flow stops, we have no proton gradient. If you have no proton gradient, then you're not going to have any ATP. If you have no ATP, you can't do any of your cell work. So all of the processes that keep you alive, you as in any organism functioning and living, all of those stop. And if your cellular machinery stops, that's the end. So we gotta have oxygen, not because oxygen helps you live, but because oxygen is the terminal electron receptor that is required to keep the proton gradient established so that we can make ATP. Be sure you know that. Practice, practice, practice. Okay. But wait, what about ATP? How does that work? Because remember, while electron flow is required for the production of ATP, electron flow is not what actually makes ATP. Right? We use electron flow to produce the proton gradient. And now, here again, the inner membrane space full of protons, very dense, highly concentrated. Our inner mitochondrial membrane, which is hydrophobic, which is why the protons can't just diffuse through and make dynamic equilibrium like it really wants to. Right? This is hydrophobic, so it can't go through. Then we have this, probably one of my favorite enzymes. If you can have a favorite enzyme, it's ATP synthase. And ACE is a common, like, hey, this is an enzyme. And ATP synthesis means we're making ATP. So the name tells us what it does. What's going to happen here is a process called oxidative phosphorylation. So remember, oxidation is losing. So who lost electrons? The reduced electron carriers in the previous part, like in the electron transport chain. That's where the oxidative comes from. So the donation of electrons and electrons moving through the electron transport chain to create this gradient is giving us the energy to phosphorylate or stick a P on it, to add a inorganic phosphate to an ADP. But it doesn't do it directly. So what's going to happen is we have created up here not just a proton gradient, but what this has done, it's like a dam. It has created an area with high potential energy. So this area has higher potential energy than the matrix, the inner part of the mitochondria. So what's going to happen is this potential energy, as the protons flow through the ATP synthase, which has a polar inside so that they can actually go through, as this flows through, it's going to turn, because this bottom part of the ATP synthase physically turns, right? It actually moves. So we're going to go from potential energy to kinetic energy. Then the kinetic energy from the ATP synthase is what powers the addition of ADP, adenosine diphosphate, with one more phosphate to become ATP, adenosine triphosphate, which is our cell currency. This is our cell energy currency. This is the actual molecule we need to make cell work happen. So the energy transfer is potential energy to kinetic energy to chemical energy, right? And so if we look back at our puzzle from before, potential energy is created when NADH releases its electrons to form the proton gradient, the oxidative part, that whole oxidation is losing. And then, so it's the proton gradient, the establishment of the proton gradient that is required to make ATP happen. The puzzle is solved, but a few big takeaways, because we still have to be able to communicate this in class to our teachers or on paper or on an exam. So, right, this whole process of NADH, a reduced electron carrier, is oxidized, right? Oxidation is losing. It loses its electrons. The electrons reduce electron receptors at the electron transport chain. That transport chain goes through a series of oxidation and reduction reactions as those electrons are passed from one to the other. The movement of electrons from a higher potential energy to a lower potential energy is going to, some energy will be released, and that's available to do cell work. What is that cell work? Establishing the proton gradient in the intermembrane space, that high potential energy area versus the matrix, which is a low potential energy area. Then, once we've established that gradient, then the potential energy, high potential energy, transforms into kinetic energy as it flows through ATP synthase from an area of high to low concentration. That's chemiosmosis. And that kinetic energy is going to put ATP or ADP and phosphate together to make ATP, which is chemical energy, our final super cell power working currency. So, yes, important things to clarify is that electrons, they don't make ATP directly, right? There's this step in between, the whole idea of the proton gradient, right? The movement, and it's not the electrons themselves. Electrons are not energy. It's the movement of electrons from one protein to another that provides the energy to form the proton gradient. And then it's the energy from the proton gradient that will produce and power the ATP production. So I know that is a lot, but suddenly it all makes sense. Oh, I love it. So, but when it comes to my AP biology course, while it is important for you to know that NADH and FADH₂ are reduced electron carriers, what you really need to be able to do, first of all, you need to be able to explain, like, you know that you actually get it if you can explain every arrow and diagram in either your book or at least in this video. So I might take a screenshot of some of these or use what you have in your book or online and try and talk through, like I just did, like all the steps. And if you can say them out loud without needing to look at notes, then you're at like the can-do level, like you get the system. But AP biology requires fluency, like the AP biology exam. Now, fluency means that if something in this pathway or one of those diagrams, if something changes, like if oxygen's not there, or what happens if there's a mutation in the fourth protein, or what happens if there's a mutation in ATP synthase, or what happens if, right? So something goes wrong, or what if something happens and the membrane goes away, right? That hydrophobic membrane. If something changes in the pathway, can you confidently predict what is going to happen downstream? And not just there will be less ATP, but you will have to be able to justify and explain why that thing that went wrong results in less ATP. So I know this is a lot. That's why YouTube has rewind and pause and rewind and pause and lets you watch a video as many times as you want. So what's really hard is that a lot of kids and students are expected to understand this in one day, with just a textbook or a PowerPoint or something that's very confusing. So I hope that this video helped explain it to you. And if it did, please do give it a like. Give it a thumbs up. Subscribe to my channel. I'm going to be doing the how electrons move in photosynthesis next. Because yes, that whole like ATP synthase, oxidative phosphorylation exists in photosynthesis as well. And I never knew that until AP biology, because that part isn't really talked about. But we're going to talk about it next. So if you want to get deep into the biochemistry, the quantitative skills, graphing skills, exam taking skills, subscribe to Miss Parrot Science. And please do leave a comment, a question, or an emoji down below. I love to hear from folks. And if you are a teacher or a student seeking extra resources to help you with things like FRQs, graph analysis, data analysis, practice with the calculations that go with the formula and equation sheet, check out my TPT store, my Teachers Pay Teachers store. It's called Know and Grow with Gnomes. Keep learning, keep growing, and we'll see you next time.